This part's going to go lickety-split because we've already done this in lab, right? This is going to be the parts of the cell. So when you look at a cell, all cells have a plasma membrane, phospholipid bilayer with a few proteins stuck in, occasional cholesterol, sometimes a little sugar tag sticking off of it. On the very most protected part of the cell, we're going to have a nucleus. But the area between the nucleus and the cell membrane is the cytoplasm. It's going to be composed of the jelly-like stuff it's a colloid, so it's mostly water, but it also has some solutes and some proteins in it, and then the organelles. We say it's gel-like. If you remember the colloid section, it could change from a liquid to more of a gel, and so that's why I say cytosol fits in the colloid section of different kinds of mixtures. Organelles are little organs. They each have their own job that keep that cell alive. Sometimes they have a phospholipid bilayer around them. Sometimes they don't. Let me say that again. Sometimes the organelles have a membrane around them, a phospholipid, just like the cell membrane. Sometimes they don't. If they have that membrane, they're called a membranous organelle, and if they don't have that membrane, they're called a non-membranous organelle. So, membranous organelles are going to be the mitochondria, the smooth and the rough endoplasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. Now, we didn't talk about the peroxisomes in lab, but we are going to talk about them in lecture. The non-membranous ones, so the ones that don't have a phospholipid bilayer surrounding them, are going to be the ribosomes, any kind of cytoskeleton, because we have different kinds of cytoskeleton, and the centrioles. Mitochondria. I don't ever want to see on an exam paper that it is the powerhouse of the cell, because when I was a baby teacher, when I was just start doing this, we were in lab, and I asked the class, "What is the mitochondria?" And they said, "Powerhouse of the cell." And I said, "Okay, what does that mean?" Crickets. Okay, so what does that mean, that it's the powerhouse of the cell? Nobody in that classroom knew that it made ATP. All they had come to college with was powerhouse of the cell, but they didn't know what that meant. Needless to say, this got under my skin. So I don't ever want to hear you say it's the powerhouse of the cell. He makes ATP. That is his job, is to make the majority of your ATP, which is what keeps you alive. Every little droplet of oxygen that you take in needs to go through a mitochondria so that it can do its job. The reason you breathe is so that that mitochondria can make ATP. He has these little folds in him. They're called cristae, and the important ATP making is happening on these. These are also phospholipid bilayer. The mitochondria itself has a double phospholipid bilayer membrane, so he is double protected, just like your nucleus. Ribosomes. This is where we're going to make some protein. So we're going to have rRNA, ribosomal RNA, that's been made by the nucleolus. And those little pieces of ribosomes are going to leave through nuclear pores and assemble in the cytoplasm. Sometimes they're attached to endoplasmic reticulum, so that would be rough endoplasmic reticulum because they have all these little bumpy ribosomes all over them. And sometimes they're free-floating. It doesn't matter where they are, they're all making proteins. It's just that if it's a free ribosome, he doesn't have to package that protein up. It doesn't have to go through the endoplasmic reticulum and through the Golgi apparatus to get packaged. He is going to be usually making proteins that that cell needs. We don't have to package it because it doesn't have to exocytose. It doesn't have to go anywhere. It's going to be used right there in that cell. Okay, so these endoplasmic reticulums that I keep mentioning, I mentioned the rough ER. There's also smooth ER, which means he doesn't have any ribosomes. These guys are tunnels, and they're actually linked to the nuclear membrane. So the rough endoplasmic reticulum, he's rough because he has all these ribosomes attached to him. He is the tunnel. Okay, so that ribosome is finished making that protein. It's going to pass through the rough ER's tunnel, and there's going to be a few little changes that are going to be made to that protein before it gets put inside of a vesicle. So goes through the endoplasmic reticulum's phospholipid bilayer and come off in a little vesicle surrounded by phospholipid bilayer, and then it's going to head to the Golgi apparatus where that vesicle is going to fuse with the Golgi's phospholipid bilayer. So unlike the rough ER that the ribosomes are attached to, those proteins are going to pass through the rough ER and it's going to modify it, maybe tag on some sort of carb or something like that, just small modifications. The smooth endoplasmic reticulum is different. He's not really working with proteins, but instead he's going to be doing things like metabolizing lipids, maybe making some cholesterol or any of the steroid-based vitamins, for example, the steroid-based hormones, maybe detoxifying some chemicals. So any sort of maybe pesticides, those are all going to be passing into your bloodstream. Be careful. Those are a different kind of solvent, not water, that can easily get into your bloodstream because they're hydrophobic. Get through those phospholipid bilayer pretty easily. Even some cancer-causing chemicals can get into there. Detoxifying drugs. You hear about the liver and the kidneys detoxifying drugs. So does it make sense that the liver and the kidney cells would have a lot of smooth ER? In the liver, when you have had, let's say, a sugar binge and you have had 12 Snickers bars, that's too much sugar. You don't need that right now. Your liver is going to store that as glycogen. When it's time to break that glycogen down into glucose, because now you've been studying for your A&P test for 12 hours and you didn't eat, that's going to happen in the smooth ER. And then, this is weird. This is different from anything that I've said so far. Inside your muscle cells, the smooth ER's got a new name. It's still smooth ER, but it's going to be called sarcoplasmic reticulum. And sarcoplasmic reticulum holds on to calcium. So when you have to have calcium to contract your muscles. Well, when it's time for your skeletal muscles or your heart muscles to contract, that sarcoplasmic reticulum is going to let go of calcium and get the ball rolling for a muscle contraction. The Golgi apparatus is next when we're talking about proteins. Well, and lipids. He's the final packager. He is the packaging plant. He is the Amazon spot where we're going to put things in a box, actually a little vesicle, but put it in a box, tag it, where is it going to go? Who bought the protein of the day? Who bought the lipid of the day? Who needs this? Who needs some cholesterol? Whatever. Who needs this? The Golgi apparatus is going to do final modifications, any sort of little extra things that might need to be tagged onto this protein or tagged onto this lipid, and it's going to squeeze it out through its phospholipid bilayer, and it's going to send out this package wrapped in its phospholipid bilayer, and it's ready to go. It's ready to leave the cell to wherever that protein or wherever that lipid needs to go. Sometimes what gets packaged up is digestive enzymes. It's also going to be put in the vesicle, but maybe that digestive enzyme needs to hang out inside that cell. So, for example, if you are a neutrophil and your job is to gobble up bacteria, to endocytose or phagocytose, right, to eat things into the neutrophil, so he's gobbled up a bacteria, that little vesicle that the bacteria is in is going to fuse with a lysosome because it's all phospholipid bilayer bound, right? They all have that little wrap around them. They're going to fuse together, and the lysosomes are going to go and kill, the enzymes inside the lysosome are going to go kill that bacteria. It's all walled off, so all of this is happening inside one big bubble. Now, honestly, most of the time, that cell has a short life. The bacteria killers, the neutrophil, so they're going to die anyway. Sometimes you have lysosomes because that cell has a short life, and the lysosome's job is actually to go ahead and kill off a cell while another nice healthy cell is going through mitosis. To lyse is to break apart. Hydrolyze, break apart with water. Lysosome breaks things apart. Digestive enzymes. Peroxisomes weren't in lab. They're actually in the model. They're little gray vacuoles. But what they do is they detoxify things. What they'll do is they will break down free radicals. So a free radical is like a charged particle that's bouncing around on our tissues that actually does damage. So what it's going to do, it's going to grab a hold of that free radical and create hydrogen peroxide. And then it's going to change it into water. Water is safe. So hydrogen peroxide, we don't want that to leak out of the peroxisome, but it's okay because once that peroxisome is done, its final product is going to be water, very safe. He does play a role in synthesis in fatty acids too, but let's just try and remember that he's detoxifying free radicals, which are damaging to our tissues. Okay, lysosomes, full of digestive enzymes. They can break down lots of different things. Maybe you've got an old mitochondria that's not making ATP anymore. Hey, that lysosome can go and break down that guy and get rid of him, but that's okay because you can make more. There are so many functions for a lysosome, but in general, he breaks things down. Sometimes, if that cell gets tired, it even breaks down that cell. This is an example of a homeostatic imbalance, Tay-Sachs disease. So what happens with this is you have some digestive enzymes that don't function properly. And when you don't have these enzymes breaking things down, then things that are supposed to be broken down build up. In Tay-Sachs disease, you get this buildup of glycolipids. So glyco, sugar, lipids, fats. And it interferes with the way your nervous system works. It's definitely a genetic disease. Central European and Jewish descent especially. And what you'll notice is that that child just doesn't he learns things, but then he doesn't progress, and slowly he actually regresses, he or she, and they die pretty young. So it's a big deal. It's a sad thing. So this is when you don't have normal digestive enzymes that are supposed to be a specific enzyme that's supposed to be breaking down glycolipids. That's that specific lysosome's job, because we have lots of different digestive enzymes.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
Okay, cytoskeleton. In Lagniappe, I have the cytoskeleton highway. Definitely a video that's cool to watch, interesting to watch, I think. And we have 3 different types of cytoskeleton. So when you think of cytoskeleton, usually we think of like a support structure, right? Skeleton, support structure. We have 3 different kinds. We have microfilaments, which are pretty thin. Intermediate filaments, they're medium size, honestly. And then we have microtubules, which are quite fat. So microfilaments, you're going to see these guys in the muscle. Maybe you've heard of actin and myosin. Well, actin is a microfilament. These guys, they can help with movement going on in the cell, actually can help change shape in the cell. Obviously, because actin definitely is one of the guys that helps you contract your muscle. You could say in your textbook, it says something about cells move when they get their actin together. Silly. Okay, but sometimes he can actually be the guy that forms that cleavage furrow that we talked about in lab. So during telophase, where you can tell now you're getting 2 daughter cells, that's going to be a actin type microfilament. Intermediate filaments are more ropey. These guys are going to attach to desmosomes. You remember the zippers that we talked about, the desmosomes? And they're going to be like cables that resist force exerting on the cell. So they are definitely creating like a toughness to the cell to keep that cell from collapsing. Microtubules are the largest of all of the cytoskeletons. In the video that I talked about, the cytoskeleton highway, you can actually see motor proteins walking across a cytoskeleton. He is likely walking across a microtubule. It's a real big, thick type of cytoskeleton. Also, those spindle fibers that you saw, those are cytoskeleton. And flagella on a sperm, or in tissues, you're going to see pseudostratified ciliated columnar epithelium. In AMP2, you're going to learn that we have ciliated tissue in the female reproductive tract. Cilia are a type of microtubule. These guys also, there's going to be linkage between different organelles. That's this guy. That's how those motor proteins like dyneins, again, watch that video, get to go walking across from one side of the cell to the other. It's a really cool video. You should watch it. So the centrosomes usually just hang out next to the nucleus until it's time for mitosis, right? And then they migrate to each of the poles. There's two centrioles, and they're at right angles to each other, and they're going to make the microtubules that you've already met. That's the spindle fibers. This is what they look like. That's very pretty. That's a pretty geometric design, yes? Here's an example of some of those other microtubules that I mentioned, the cilia and the flagella. So sperm, those little tails coming off of it. The tails are flagella microtubules. When we do the respiratory tree, in AMP2, we'll look at these little cilia coming off of the respiratory tissues of the trachea. Those little cilias are what die when you smoke, and therefore you get a cough. Because these cilia, their job is to catch dust particles and whatever before it gets down to the alveoli where you have oxygen carbon dioxide exchange. That's bad. We don't need anything down there but air. These guys catch it and help bring those things up your airway to your throat so you can swallow it and put it in your stomach. But if you smoke, you kill these guys. Stop smoking. Here's another example of a type of microtubule, microvilli. These are going to be found in different areas. They're in the kidneys, but the one that we harp on a lot is in the GI tract. So along here, this is all going to be phospholipid bilayer, and little proteins embedded so that you can absorb your food. Okay, the nucleus, huge important guy because he contains DNA. DNA is what makes you, and it must be very protected. So he actually has a double phospholipid bilayer, just like the mitochondria. Most cells just have one nucleus. Red blood cells don't have any, but because he doesn't have a nucleus, he has a short life. Some cells have lots and lots of nuclei because of the way they're formed in utero. They actually fuse together. So their skeletal muscle is loaded with nuclei. Nuclei is plural for nucleus. This picture highlighting the nucleus. So this is just the nucleus. The cell, rest of the cell's been taken away. This is a nucleolus. That's a different organelle. The nucleolus is living inside the nucleus. He makes pieces of ribosomes, ribosomal pieces, ribosomal subunits, ribosomal RNA. The nucleus has got a double phospholipid bilayer, and then it has these little pores so that things can pass out of the nucleus or into the nucleus, but there's rules on that, and they're going to be really tiny holes. And of course, all throughout the nucleus, we have our DNA. As long as we are just in interphase, the cell's just doing its job, it's in chromatin form. This is a picture of that double phospholipid bilayer. So this goes all the way around the nucleus. This is showing a pore, but there's a phospholipid bilayer, and looky there, there's a phospholipid bilayer. This and this are going to go all the way around that nucleus, all the way. We're going to protect that DNA. Nucleoli, again, making ribosomal subunits. There's usually just one or two per cell. This picture is, you don't want this picture to be part of your life. This is lymphosarcoma. 1, 2, well, let's start with this. This is just one, although I see a second one here, but they're not the same size. 1, 2, 3, 4. This has 2 nuclei, not normal. So 1 nucleus, 2 nucleus, and they each have a nuclei in it. These guys didn't go through mitosis properly, Why? Because it's cancer. He's dividing too fast and not paying attention to what he needs to be doing. But whenever you have cancer, it makes for a beautiful slide to show off what a nucleoli looks like. So more than one nucleus, one nucleolus, multiple nucleoli. So this is nucleolus, this is nucleolus, this is nucleolus, this is nucleolus, this is nucleolus. Multiple nucleoli in there. Okay, chromatin and chromosomes. Did this in lab. Chromatin is thin. This is how our DNA is 99% of the time. It's usable DNA. This right here has been all stretched out, but there's sections of these nucleotides right here that make up a gene. So this DNA that's all the way up to this bundle of a chromosome, this DNA has thousands of genes for all kinds of things in your body. Chromatin, to create a new protein, you need chromatin so that you can read all the C's, G's, A's, T's. Because when you're making a protein, you need to be able to read the gene. So let's just say this is one gene right here. This A all the way through this A. You got to be able to read that to make whatever protein that gene is coding for. When it's time to go through mitosis, chromatin, which is thin, gets really squeezed up into a chromosome. So one chromosome is actually a stick. So I wish this wasn't drawn as an X because one chromosome is a stick. But when it's time to go through mitosis, you have to double the DNA, so you get two sisters, one stick, two stick, and they're stuck together by something called a centromere.